1.4 Post-fertilization structures and events. Following double fertilization, events of endosperm and embryo development, maturation of ovule into seed and ovary into fruit are collectively termed post-fertilization events. 1.4.1 Endosperm. Endosperm development precedes embryo development. Why? The primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. The cells of this tissue are filled with reserved food materials and are used for the nutrition of the developing embryo. In the most common type of endosperm development, the PEN undergoes successive nuclear divisions to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water form tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, example pea, groundnut, beans, before seed maturation, or it may persist in the mature seed, example castor and coconut, and be used up during seed germination. A split opens some seeds of castor, peas, beans, groundnut, fruit of coconut, and look for the endosperm in each case. Find out whether the endosperm is persistent in cereals, wheat, rice, and maize. 1.4.2 Embryo. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Though the seeds differ greatly, the early stages of embryo development, embryogeny, are similar in both monocotyledons and dicotyledons. Depicts the stages of embryogeny in a dicotyledonous embryo. The zygote gives rise to the proembryo and subsequently to the globular, heart-shaped and mature embryo. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledons is the epicotyle, which terminates with the plumule or the stem tip. The cylindrical portion below the level of cotyledons is hypocotyle that terminates at its lower end in the radicle or root tip. The root tip is covered with a root cap. Embryos of monocotyledons possess only one cotyledon. In the grass family, the cotyledon is called a scutellum that is situated towards one side lateral of the embryonal axis. As it lowers in, the embryonal axis has the radicle and root cap enclosed in an undifferentiated sheath called coleorrhiza. The portion of the embryonal axis above the level of attachment of scutellum is the epicotyle. Epicotyle has a shoot apex and few leaf primordia enclosed in a hollow foliar structure, the coleoptile. Soak a few seeds in water, say of wheat, maize, pea, chickpeas, groundnut, overnight. Then split the seeds and observe the various parts of the embryo and the seed. 1.4.3 Seed. In angiosperm, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. Seeds are formed inside fruits. A seed typically consists of seed coat, cotyledon, and an embryo axis. The cotyledons of the embryo are simple structures, generally thick and swollen due to storage of food reserves, as in legumes. Mature seeds may be non-albuminous or ex-albuminous. Non-albuminous seeds have no residual endosperm as it is completely consumed during embryo development, that is pea, groundnut. Albuminous seeds retain a part of endosperm as it is not completely used up during embryo development, that is wheat, maize, barley, castor. Occasionally in some seeds such as black pepper and beet, remnants of nucellus are also persistent. This residual persistent nucellus is the perisperm. Integuments of ovules harden as tough protective seed coats. The micropyle remains as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. As the seed matures, its water content is reduced and seeds become relatively dry, 10 to 15% moisture by mass. The general metabolic activity of the embryo slows down. The embryo may enter a state of inactivity called dormancy, or if favorable conditions are available, adequate moisture, oxygen, and suitable temperature, they germinate. As ovules mature into seeds, the ovary develops into a fruit. That is, the transformation of ovules into seeds and ovary into fruit proceeds simultaneously. The wall of the ovary develops into the wall of fruit called pericarp. The fruits may be fleshy as in guava, orange, mango, etc., or may be dry as in groundnut and mustard, etc. Many fruits have evolved mechanisms of dispersal of seeds. Recall the classification of fruits and their dispersal mechanism that you have studied in an earlier class. Is there any relationship between number of ovules in an ovary and the number of seeds present in a fruit? In most plants, by the time the fruit develops from the ovary, other floral parts generate and fall off. However, in a few species such as apple, strawberry, cashew, etc., the thalamus also contributes to fruit formation. Such fruits are called false fruits. Most fruits, however, develop only from the ovary and are called true fruits. Although in most of the species, fruits are the results of fertilization, there are a few species in which fruits develop without fertilization. Such fruits are called parthenocarpic fruits. Banana is one such example. Parthenocarpic carpi can be induced through the application of growth hormones, and such fruits are seedless. Seeds offer several advantages to angiosperms. Firstly, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seeds have better adaptive strategies for dispersal to new habitats and have the species to colonize in other areas. As they have sufficient food reserves, young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection to the young embryo. Being products of sexual reproduction, they generate new genetic combinations leading to variations. Seed is the basis of our agriculture. Dehydration and dormancy of mature seeds are crucial for a storage of seeds, which can be used as food throughout the year and also to raise crop in the next season. Can you imagine agriculture in the absence of seeds, or in the presence of seeds which germinate straight away soon after formation and cannot be stored? How long do the seeds remain alive after they are dispersed? This period again varies greatly. In a few species, the seeds lose viability within a few months. Seeds of a large number of species live for several years. Some seeds can remain alive for hundreds of years. There are several records of very old yet viable seeds. The oldest is that of a lupine, Lupinus arcticus, excavated from Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. A recent record of 2,000 years old viable seed is of the date palm, Phoenix dactylifera, discovered during the archaeological excavation at King Herod's palace near the Dead Sea. After completing a brief account of sexual reproduction of flowering plants, it would be worth attempting to comprehend the enormous reproductive capacity of some flowering plants by asking the following questions. How many eggs are present in an embryo sac? How many embryo sacs are present in an ovule? How many ovules are present in an ovary? How many ovaries are present in a typical flower? How many flowers are present on a tree? And so on. Can you think of some plants in which fruits contain very large number of seeds? Orchid fruits are one such category, and each fruit contains thousands of tiny seeds. Similar is the case in fruits of some parasitic species such as Orobanche and Striga. Have you seen a teeny seed of ficus? How large is the tree of ficus developed from that teeny seed? How many billions of seeds does each ficus tree produce? Can you imagine any other example in which such a teeny structure can produce such a large biomass over the years? 1.5. Apomixis and polyembryony. Although seeds in general are the products of fertilization, a few flowering plants such as some species of Asteraceae and grasses have evolved a special mechanism to produce seeds without fertilization called apomixis. What is fruit production without fertilization called? Thus, apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reduction division and develops into the embryo without fertilization. More often, as in many citrus and mango varieties, some of the nuclear cells surrounding the embryo sac start dividing, protrude into the embryo sac, and develop into the embryos. In such a species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is referred to as polyembryony. Take out some seeds of orange and squeeze them. Observe the many embryos of different sizes and shapes from each seed. Count the number of embryos in each seed. What would be the genetic nature of apomicted embryos? Can they be called clones? Hybrid varieties of several of our food and vegetable crops are being extensively cultivated. Cultivation of hybrids has tremendously increased productivity. One of the problems of hybrids is that hybrid seeds have to be produced every year. If the seeds collected from hybrids are sown, the plants in the progeny will segregated and do not maintain hybrid characters. Production of hybrid seeds is costly and hence the cost of hybrid seeds become too expensive for the farmers. If these hybrids are made into apomixed, there is no segregation of characters in the hybrid progeny. Then the farmers can keep on using the hybrid seeds to raise new crop year after year and he does not have to buy hybrid seeds every year. Because of the importance of apomixis in hybrid seed industry, active research is going on in many laboratories around the world to understand the genetics of apomixis and to transfer apomictic genes into hybrid varieties. Summary. Flowers are the seat of sexual reproduction in angiosperm. In the flower, androecium consisting of stamens represent the male reproductive organs and gynoecium consisting of pistil represent the female reproductive organs. A typical anther is bilobed, dithecous and tetrasporangiate. Pollen grains develop inside the microsporangia. Four wall layers, the epidermis, endothecium, middle layers, and the tapetum surround the microsporangium. Cells of the sporangious tissue lying in the center of the microsporangium undergo meiosis microsporogenesis to form tetrads of microspores. Individual microspores mature into pollen grains. Pollen grains represent the male gametophytic generation. The pollen grains have a two-layer wall, the outer exine and inner intine. The exine is made up of a sporopollenin and has germ pores. Pollen grains may have two cells, a vegetative cell and generative cell, or three cells, a vegetative cell and two male gametes at the time of seeding. The pistil has three parts, the stigma, style and the ovary. Ovules are present in the ovary. The ovules have a stalk called funicle, protective integument and an opening called micropyle. The central tissue is the nucellus in which the archisporium differentiates. A cell of the archisporium, the megaspore mother cell divides meiotically and one of the megaspores form the embryo sac, the female gametophyte. The mature embryo sac is 7-celled and 8-nucleate. At the micropylar end is the egg apparatus consisting of 2 synergids and an egg. At the chalazal end are 3 antipodals. At the center is a large central cell with 2 polar nuclei. Pollination is the mechanism to transfer pollen grains from the anther to the stigma. Pollinating agents are either abiotic, wind and water, or biotic animals. Pollen-pistil interaction involves all events from the landing of pollen grains on the stigma until the pollen tube enters the embryo sac when the pollen is compatible, or pollen inhibition when the pollen is incompatible. Following compatible pollination, pollen grain germinates on the stigma and the resulting pollen tube grow through the style, enter the ovules and finally discharge 2 male gametes in one of the synergids. Angiosperm exhibit double fertilization because 2 fusion events occur in each embryo sac, namely syngamy and triple fusion. The products of these fusions are the diploid zygote and the triploid primary endosperm nucleus in the primary endosperm cell. Zygote develop into the embryo and the primary endosperm cell form the endosperm tissue. Formation of endosperm always precedes development of the embryo. The developing embryo passes through different stages such as the pro-embryo, globular and heart-shaped stages before maturation. Mature dicotyledonous embryo has two cotyledons and an embryonal axis with epicotyle and hypocotyle. Embryos of monocotyledons have a single cotyledon. After fertilization, ovary develops into fruit and ovules develop into seeds. A phenomena called apomixis is found in some angiosperm, particularly in grasses. It results in the formation of seeds without fertilization. Apomixes have several advantages in horticulture and agriculture. Some angiosperms produce more than one embryo in their seed. This phenomena is called polyembryony. आज आपका चैप्टर एंड हुआ। एनसीईआरटी ऑडियो बुक को आप एनसीईआरटी लाइन बाय लाइन सुनकर याद रख सकते हैं। इस चैप्टर को 4 पार्ट में रिकॉर्ड किया गया है। इससे आपको पार्ट वाइज सुनने में आसानी होगी और जो पार्ट आपकी वीक है उसको आप कम समय में बार बार सुन पाएंगे। विशेष चैप्टर को कॉन्सेप्चुअल तरीके से समझने के लिए आप हमारे चैनल के वीडियो में जाकर देख सकते हैं। थैंक यू।